वेलकम. वेलकम. हो सर.
चलो ऑन द बेस्ट. Yes, madam. On the best. Yeah, actually, we come to the best.
वेलकम ची स्टेट मॅडम यूट्यूब लाईक करा.
अंजू ते हे कर ना डीना किंवा बी बॉल.
वेस्ट एक लॅग येईल. डन मॅडम. व्हेरी गुड.
सर हे पण
मॅडम ऐका ना 90 झाले ना 90 डी नाही करायचं. तेच आपल्याकडे प्रोग्राम
नंबर दिसतो.
हा 50 का डायरेक्ट आपला व्हिडिओ. बॅकअप मध्ये तुमचे मोबाईल नीट आहे. आता सध्या नेट तुमचं लॅनवर चालू आहे बरोबर.
लॅनवरच
आणि तुमचं सरांचं ईमेल आयडी लॉग इन केलंय त्याचे.
झालं सर हे पण येतात. लाईव्ह चॅट पण स्टार्ट झाले. अच्छा तिथे मेसेज टाकतात वाटतं ते याच्यामध्ये.
थँक्यू सर.
6 मेंबर आहेत सर यूट्यूबला. चेक नाही करतात. झालं सर मोठंच.
काय सांगायला होतं मॅडम 90 आहे ना की ॲडमिशन
पुढे जातच नाही.
त्यांना काही थोडी दिसते किती
आमचा बोनस होतो ना आम्ही काय
तो मेसेज जाऊ नका. लोकांना हे कळेल की गुगल मीट ऑलरेडी फुल झाले म्हणून डिनाय करत आपण मग कशाच करू.
आपण सांगायचं ना गुगल मीट सोडून काय
असं
तरी पण वाचत नाहीत ना कोणी.
ट्राय करतात एखादा फोन बी करायचं तुमचं जवळ झालं ना सर लगेच तुम्ही सांगा काइंडली ड्रॉइंग ऑन द बोलते
38 झाले.
100 ते तरी भरत वाटतं कारण 790 आहे वाटतं त्याच्यापैकी अर्ध जेवढं दिलं ते बघा अर्ध जास्त नाही होणार
युट्यूबला
काय आपल्याला
अच्छा
पैसे दिले
आम्ही कुठं
याचे पैसे लय खर्च करतो
आम्ही म्हणत होतो झूम वर घ्या. आम्ही
आम्ही
गेम
तुम्हाला
नाही
म्हटलं
केला.
तुम्ही मनावर नाही गेले
कॉलेज
सरांनी मनावर घेतलं तर काय मला नाही बघा मॅडम जे म्हणून विठ्ठल सरांनी फक्त मनावर घ्या आणि आज तुमचा दिवस आहे खास हा? नाही का मॅडम आषाढी आहे हो मॅडम
आपलं बनवलेले शर्ट पॅन्ट
विठ्ठलाची डिमांड वाढल्यावर विठ्ठलाची डिमांड वाढल्या
सर आपलं याला पण युट्यूबला पण चालू झालं.
जॉईन करायचं
आहेत 5 जण.
शंभर दिले त्याच्यापासून शिकलो काय आपली अपेक्षा 100 होती गुगल मीटची
जास्त झाले तर चांगलंच आहे सर.
साऊथ नॉर्थ सगळे काही काढून करायचं आपण कॉलेजचं नाव ऑल इंडिया झालं टॉपिक इतकं छान असतं लोक जॉईन होतात. येईल मॅडम प्रिंट स्क्रीन करत राहा बरं फोटोग्राफ शेवटी
स्पीकर बोलताना वगैरे.
तुम्ही लास्ट बोलावा ना सर
तुम्ही तशी एक अनाउन्समेंट करा ना. व्हिडिओ.
चलो फ्रेंड्स
जैसे
ऑल द बेस्ट मॅडम
येस सर.
मोहनतीचं झाल्यानंतर लगेच गोरे मॅडम बोलणार आहे.
आपण क्लासला कॉलेज टू
स्टार्ट विथ द कॉलेज टू
आणि कॉलेज टू संपला की इमिडिएटली मूव करायचं प्रेझेंटेशन. वेलकमची स्लाइड.
वेलकमची नाही डायरेक्ट वॉटर स्टुडंट
ते आता इथे नाही होतं सर. पीपीटी ऑनलाईन चेंज नाही करत. ते ज्याकडे जसं मी हे केलं ना स्क्रोल अप केली ना प्रेझेंटेशन तसं करावं. कारण पीपीटीचा आपण सेपरेट टॅब ओपन केलाय ना तो शेअर केला जातो. ॲप शेअर होतो ना तोच डायरेक्ट डिस्प्ले झाला. आपल्या तिकडे तेच सेपरेट टॅब ओपन केला त्याच्यावरच स्क्रोल डाऊन करायचं किंवा बंद करताना बॅनर डिस्प्ले करावं लागेल आणि मग स्टॉप दिले.
तुम्हाला हे स्टॉप शेअर करावंच लागेल.
हा मग स्टॉप शेअर केल्यावर आपण एक स्लाइड पुढे घेऊन करू शकतो का? करू शकतो ना.
थोडंसं स्क्रोल जरी झालं
तरी काय
काय
हो ते आधी आपण वापरायचं.
बरं मॅडम साधारणतः आपल्याला 11:15 लिहिलं पाहिजे.
तेच येणार आहे का खालूनच करायचं?
खालून नका करू म्हणा कारण की वेळेवर जॉईन होत
आले असतील तर नंबर काहीतरी या.
हां आणि 100ची आपण संख्या ठेवू. लॉगिन जर नाही झालं त्या व्हिडिओवर तर लाईव्ह जायचं.
आलाय तुम्ही बघितला? स्क्रिप्ट बनवतो. गाडीच नाही आवडत पण इकडे येऊन बोला ना तुम्ही. बघ मग तेच कन्फर्म करायचं होतं. चालेल. सोपं पडेल हां त्याच्यामुळे चालेल बोलूया. चालेल चालेल या. इकडे म्हणता म्हणजे मी जे बोलणार आहे ना बदलाचं थोडी मी पहिल्यांदा बसायला मध्ये वेळ लागणार ते काय टाकशील बॅनर ते ते तुम्ही म्हणायचं लाइटिंग ऑनचं ते डिस्प्ले करायचं आपण इकडे कमी करायचं नाही ते आणि रिटर्न मुकेला असायचे स्पीकर असायचे म्हणजे ते परत म्हणायचं व्हेरी प्लेजरिंग ये अगं माझं पेपर आहे मी ते शुभमला ऑपरेट करायला सांगते पेपर कंडक्ट करायला सांगते त्याला जमेल का बरं तुम्ही त्याला पहिला
VM जमत नाही त्याला
उजवीला असं ना थोडं
एक्सलेंट कनेक्शन नंतर
40 झाले
हो
नाही व्हिडिओ 40 50 इनको मानसराज विश्व इधर मीटिंग दोघांनी सुरू हॅलो शुभम ऐक ना मी तुला माझा व्हिडिओचा आयडी पासवर्ड पाठवते त्याच्यावर मीटिंग शेड्यूल
वेळ नाही
वेळ ती मीटिंग तुला करता येईल का म्हणजे एक्झाम होईल फक्त ठीक आहे तुला आयडी पासवर्ड पाठवते आणि नंतर महिन्याचे क्वेश्चन ही बॉटल माझी का
काय माहित इकडेच राहिले असेल कदाचित म्हणजे ऍडमिट करायचं ना सगळ्यांना नंतर घेणार म्हणा मी आजच नाही फक्त दुपारी टाकते म्हणा असाइनमेंट दुपारला टाकून दे अरे माझं नेट खरं वेगळं तर एक्सलेंट कनेक्शन म्हणते सर यूट्यूबची ती लिंक क्लिअर आहे का बघा ना कारण की इथं एक्सलेंट कनेक्शन दाखवतंय पण माझ्या ह्याच्यात असं ब्लर दिसतंय थोडंसं
किती वाजेपर्यंत आहे आपला प्रोग्राम? एक पर्यंत म्हणजे 2चा टायमिंग देते. 2 PM.
क्लिअर आहे ना सर? तर मग माझ्या नेटचा काहीतरी प्रॉब्लेम असेल.
एकदम मस्त दिसतं.
क्लिअर आहे ना? कारण की इकडून एक्सलेंट कनेक्शन आहे.
याच्यावर हे मायक्रोफोनचं चिन्ह काय आहे?
मायक्रोफोन?
युट्यूबवर
मायक्रोफोन अच्छा ते तिथे मध्ये सर ते चोपडे सरांचंच आहे ना हे आपण त्याच्यामुळे येतोय.
हे बघा हे इथं आलं.
आता नाही येत ते कर्सर फिरवला की येत आहे.
अच्छा
आता गेलं ना?
गेलं.
ते कर्सर सर फिरवला की ते येत आहे.
चला ऑल सेट.
तुला फुल स्क्रीन दिसते. मी शुभमला बिहेवियर माझ्या आर्टिकलचं ऑप्शन करायला सांगितलं तर फक्त त्यांना जर काही प्रॉब्लेम आला ना तर बघू आता ते स्टार्ट तुम्ही करा ना.
सगळ्यांचा मोबाईल सायलेंट करून ठेवा.
सायलेंट करून ठेवा फक्त.
केलाय मी.
मॅडम 10:50ला व्हिडिओ झाला मॅडमकडे आणि मॅडमचं झालं की मोठी स्लाईड आहे आणि मॅडम जेव्हा बोलतील ना तेव्हा ती वॉटर सिग्नेचर बॅनर आहे ते डिस्प्लेला राहतं.
ते ब्लू कलरचं.
हां वेलकम स्लाईड
कॉन्फरन्सच झालं.
हां कॉन्फरन्स ते वेबिनारचं बॅनर.
वेलकम झाल्यावर जे स्लाईड ठेवले ना त्यावेळेस मॅडम बोलत राहते.
एक वर करशील कसं बरं कधी लक्ष
ओके चलो ऑल द बेस्ट.
सर एक दोन मिनिटं आहेत का? मी फक्त ते सेटअप करून ठेवा. ठीक आहे मग.
Please can you
ग्रुपवरच मेसेज टाकले की तुम्ही काही वेगळे असेल तर शुभम म्हणजे मला कोण येणार शुभम
तेच म्हणते मी फर्स्ट इयर वाल्यांना माहित नाही शुभम.
त्यांना नंबर दिला मी त्यांना आणि मग डायरेक्ट मग मेडिकलच्या वेळेला सबमिशनला तर शुभम
सर प्ले करून टाका व्हिडिओ. तर अनम्यूट करा फक्त.
मॅडमला सांग इको येईल. Good morning, sir.
Hello, Dr. Vikram sir. Good morning.
Good morning, Pratap sir. We can hear you.
Okay.
A very good morning to everyone present here with warm greetings. Very good morning. I, Professor Rupali Jinturkar, feel privileged to welcome all the learned delegates, respected teachers, PhD scholars, and students. Dear participants, the upsurge of technology has revolutionized our lives. In pharma field, to upgrade the research, use of sophisticated and automated instruments for better accuracy and advanced separation is a need of time. And considering this fact, we at Modern College of Pharmacy Nigdi Pune has organized this webinar. It gives me immense pleasure to extend warm welcome in one-day national webinar entitled Water's QDA Mass Detector with CAMAG HPTLC MS Interface Technique on behalf of convener and principal Dr. P.D. Chaudhary and coordinator Dr. V.V. Chopade, Associate Professor, Progressive Education Societies, Modern College of Pharmacy, Nigdi Pune. We want to express our heartfelt thanks to Waters India Private Limited, Mumbai, for associating with us for this webinar. Now, I request Coordinator Dr. V.V. Chopade to deliver welcome speech and brief out objectives of this webinar.
Thank you, madam. Thank you, Rupali madam. Am I audible very
Yes,
clearly to everyone? Very
Yes, sir.
good morning to Monty sir. Monty sir is there? Hello? Prakash Monty sir is there?
Yeah. He joined the meeting.
Yes, sir. I am
Sir,
available.
sir.
I am available, doctor.
Good morning, sir.
Good morning.
Good morning, sir. Very good, sir. Thank you, sir. Thank you, sir. So, my dear colleagues and friends, very good morning to one and all. I, Dr. Vittal Chopade, coordinator of this national level webinar on Waters QDA mass detector with CAMAG HPTL MS interface technique for pharmaceutical analysis in the association with our Waters India Private Limited, Mumbai on 20 July 2021. On the behalf of management, principal, and all faculty of Progressive Education Society, Modern College of Pharmacy, Yamuna Nagar, Nigdi Pune, I heartily welcome you all for this webinar. Friends, society, College of Pharmacy is a premium institute education in the pharmaceutical sciences in Pune, March 1998. Our college offers pharmacy education in the basic undergraduate course like B.Pharm with 100 intake, M.Pharm in 4 disciplines with intake 42, PharmD with 30 intakes. The college is also recognized as a PhD research center by Savitribai Phule, Pune University in the Department of Pharmaceutical Sciences. I am happy to share that total 44 research scholars have enrolled for their doctoral study and 10 students awarded with PhD from our it is a matter of for us. The college is approved under section. Madam, I am audible clearly? Rupali ma'am?
Yes, sir.
Hello?
Yes, sir.
Yes, sir, you are
audible.
audible, sir.
Okay. So it is a matter of great pride for us. The college is approved under section 2F and 12B Act UGC 1956. The college is accredited by NAC as well as the NBA. The college has covered a special ability in the imparting the excellent education which is reflected by the NIRF ranking, that is 41st rank in 2017, 74th rank in 2019 and 68th one rank in 2020. I am proud to state that among 35 faculty members of our institutes, 23 have successfully completed their PhD. Also, as a part of best teaching and learning practices, more than 70 students have secured the position in top rank in Savitribai Phule Pune University, involved the two gold medalists. The college is ponders to promote a research culture among its stakeholders, and in this context, faculty members have generated a research grant worth rupees 1.84 crores from various national and international funding agencies. Uh, Friends, I am happy to share that our faculty members have completed and was working on the 5 major and 37 minor research projects under the guidance of Dr. P.D. Chaudhary sir at the well-equipped research laboratory of Modern College of Pharmacy in Nigdi Pune. The Savitribai Phule Pune has appreciated the holistic development and achievement of institutes and has awarded the prestigious Best Professional College Award in 2015 and 16. Friends, this college taking consistent and concerted efforts for holistic development of our students and faculty. Moreover, the college provides ideal forum to bring together for pharmacy teachers, researchers, and industrial experts, as well as the PhD scholar or inter and multidisciplinary sector to discuss, deliberate, and interact to share their views on the topic of current interest in order to explore the new vistas. So, friends, we know we are suffering from the COVID pandemic situation nowadays. So, current COVID pandemic situation have learned to us how to live without coming together, how to interact with you online by using the online platform. So, provide us and the opportunity to extend it's to come together at a very large level. So it's give me an immense pleasure to offer my fellow colleagues, research scholars, pharmacy teachers, professionals, industrial persons, welcome in national level webinar on Waters QDA mass detectors with CAMAC HPLC mass interface techniques for pharmaceutical analysis with the objective of introduce the fundamental TLC and HPLC mass concept, explain the function of each major component of TLC and HPLC MS systems. It indicates the major advantage and TLC and HPLC MS, the applications used for the pharmaceutical active ingredient as well as the synthetic drugs, also the uh, national sources component. So, friends, peace, it's organized by the Peace Society Modern College of Pharmacy Nigdi Pune will reach you all with the modern and innovative techniques with develop of the research skill. So I'm confident that this webinar will be certainly update and upgrade you the technical skill with the reference to research and the teaching learning process. So I'm thankful to Mr. Prakash Monti sir, Senior Marketing Manager, Waters India Private Limited Mumbai, as well as Dr. Ritesh Oza sir, Production Specialist, Water India Private Limited Mumbai, for accepting our the invitation in very short time for sparing their valuable time of this national level webinar. So I'm thankful to Honorable Dr. G.R. Ekbote sir, Chairman, Business Council, Progressive Education Society, Professor Shamkant Deshmukh sir, Honorable Secretary, PE Society Pune, all Business Council member, and Dr. P.D. Chaudhary sir, Principal and Convener of this national level webinar, for their continuous guidance, their constant support, valuable encouragement for development of our institute, as well as the organize this type of the webinar and conferences. I wish all the very best to the participant and successfully completion of this one-day national level webinar on Waters QDA mass detector with CAMAC HPTLC mass interface technique for the pharmaceutical analysis. Last but not least, I'm very thankful to all teachers, researchers, and PhD scholars who have been participate in this national level webinar. I pray my sincere thanks to all HODs, principals, and principals of pharmacy colleges from all over India. So last but not least, I'm very much thankful to all researchers, teachers who have participated in this webinar. Also, I pay my sincere thanks to all HODs, principal of pharmacy colleges from all over India who have deputed their faculty and research scholar for attending this national level webinar. Okay, so I wish all the best. Keep learning and keep upgrading yourself. Okay, thank you very much to one and all. Now I hand over to session Professor Rupali Jinturke ma'am. Thank you, ma'am.
Yes, sir.
Thank you so
Thank you
much.
so much.
Now
Now let's welcome our today's chief guest,
Mr.
Mr. Prakash Mohanty. Dear participants,
It's
it's
our
our
holy
holy
tradition
tradition to worship our goddess Saraswati
before
before
any
any
important
important
event.
event.
So
So let's dedicate a prayer to Ma
Ma
Saraswati. I
I
request our chief guest to inaugurate the webinar with lamp lighting.
वस्त्रावृता या वीणा वरदण्डमण्डितकरा या श्वेत पद्मासना या ब्रह्माच्युत शङ्कर प्रभृतिभिः देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेष जाड्यापहा.
Welcome, sir. Welcome to Dr. Prakash Mohanty on behalf of Modern College of Pharmacy, Nigdi Pune. I request our chief guest to kindly accept virtual bouquet on behalf of convener and principal Dr. P.D. Chaudhary, sir.
Thank you, Doctor. Thank you, Dr. Victor. Thank you, Dr. Vipali.
Yes, sir.
Dear
Dear participants, I would like to give brief introduction about our chief guest. Dr. Prakash Mohanty is currently working as Senior Marketing Manager, Waters India Private Limited, Mumbai. He has more than 26 years of experience in sales and marketing of sophisticated analytical and laboratory instrumentation techniques catering to various industries. He is associated with Waters India, Mumbai, and is currently responsible for marketing Waters products and technologies in Western region, primarily to pharmaceutical industries, academic institutes, and research labs as well. By qualification and training, he is an engineer with MBA in marketing from XIMB Bhubaneswar. We are happy to introduce and welcome him to our forum. We request him to present us with Waters Corporation overview, new technologies and products for various market verticals, benefits of Waters solutions that can improve lab productivity, regulatory compliance and data management in pharma research or QC labs. Now I request our chief guest Mr. Prakash Mohanty to address the participants on topic benefits of water solutions that can improve research productivity. Over to you, sir.
Thank you, Dr. Rupali, and thank you, Dr. Vittal, for honoring me and giving me an opportunity to address the august presence of all over India, the scientists from the pharmacy colleges and the professors, the principals, and the various students. They have participated in this event. They are the future analysts. They are the scientists on whose hand the equipments will be given for performance. The equipments will be given to produce results. So it is indeed a pleasure to be present here to give a brief talk. And I'll just open my slide. Give me a second.
So there is a little background noise at your side.
Oh yeah, actually, ma'am, there is a construction going behind my home. We are actually at work from home. And, it's a huge.
Okay, sir. Okay. Okay, sir. So you have to please speak a little louder because you are not audible sometimes because of that noise.
Yeah, can you see my screen, please?
Yes, sir. I can.
Okay. So thank you so much, Dr. Rupali, and thank you, Dr. Vittal sir, for inviting us to this event. And I am honored indeed to be the guest of honor today. And it's my pleasure to tell you that it all started with a humble request from my colleague Padmanath to Dr. Vittal sir to address all India faculty, all India students on this platform today. And I am very happy to be here as a chief guest to give an orientation to all my colleagues, those who are in this particular event. I mean, colleague means every one of you who are here. So I will first of all thank Dr. P.D. Chaudhary, the principal of Modern College of Pharmacy, and also Dr. G.R. Ekbote, the chairman, sir, to be present here. And their presence and their kind courtesy in allowing us to give a nice talk on enabling technology QDA mass detector. To all of you, dear students, will be definitely helpful to you in your career ahead. And to all the scientists, all the participants, definitely this is an enabling technology which can help you. Okay, so with this, let me go ahead and my colleague Dr. Ritesh Ojha will be the speaker today. And earlier during our initial invitation process, we had mentioned about Dr. Ketan Savan, but he fell sick yesterday. He had dengue, and my sincere apology to all of you. But my colleague Dr. Ritesh is a competent authority in this subject, and he is knowledgeable, experienced. He will definitely do a very nice job in terms of explaining you about this technology. So with this, I will start the presentation from my side, which is on giving you an idea about what's happening around the world, what Waters is doing for the scientific fraternity across the globe. We started as a company in about 1958 by the founder, Jim Waters. That's how the name is, Waters. And during the journey of last 65 plus years, we have been introducing various technologies focused on LC, LCMS, separation science, and also the data and the consumables related to that. So we are very focused company, specific product line. We don't have an entire product basket which you may need for your research activity in the laboratory. So when we are focused, obviously we are trying to address the scientific challenges, industry challenges, and the researchers who are always looking for better sensitivity, higher efficiency in the separation, and also the lower limits of detection and quantitation as well. So what I am trying to highlight you a few products that has made the landmark in our journey. One is the Empower software, which is today, everything is digital. Even our event is happening digital, virtual. So the software has made all the differences because Indian companies, all of them are facing US FDA audits. Compliance is the key priority for them. Data integrity speaks a lot about their preparedness for the audit. And now coming to the other enabling technology, in 2004 we introduced a product called UPLC. All of you are familiar with HPLC, high performance liquid chromatography. From high performance to ultra performance, which is Waters product, and we first launched it. It's a patented product by Waters, and then we hold a huge market share for this. And this product gave three significant advantages to our customers. One, they were looking for speed of analysis. Second, they were looking for sensitivity and better resolution. I mean, the better efficiency while separating. All three in one product. And Equity QDA, today Dr. Ritesh will explain you how this particular mass detector is coupled with HPTLC MS interface and what are the various scopes it has to help you in your research program. We are a company who is certainly business oriented because we have to produce results. We are a public listed company in New York Stock Exchange, and globally there are investors who are looking at Waters in terms of the performance year on year, quarter on quarter, and the growth. In addition to that, the growth is important, but at the same time, Waters always brings newer technologies to satisfy our customers' needs or capabilities they are looking for as a company. When you are an innovator, you are always challenged by the scientific community because they expect you to solve their problem. So Waters has too many scientists working across the globe, various R&D centers, various regional laboratories in terms of producing equipments that can be directly applied to the customer applications. And that is where we get the satisfaction when we solve your problem. We are there with you, we train you, we make you successful. So we are a company which is about $2.2 billion sales, and the addressable total market globally is about $11 billion. So we have a good market share and we have been doing good. 9% of our revenue goes into R&D spending. That means you can trust Waters will be bringing newer technologies, and we look forward to collaborate with all of you anytime when you face a challenge. We are welcome and you can collaborate with us, we can work together. These are the 5 different product lines that Waters focuses. First is the separation science, which means LC, HPLC, UPLC, and UHPLC, and then the preparative HPLCs for purification, impurity isolations, and of course there is one more technique called SFC, which is supercritical fluid chromatography that works on carbon dioxide, which is in supercritical stage, as one of the major source of solvent, and it is added with some 5 to 10% organics, maybe ethanol, methanol, so that it can give a good selectivity. Then comes the mass spectrometry. Dr. Ritesh is the most competent authority to explain you these products. We start with single quad mass detector QDA, then tandem quads, which is MSMS for fragmentation. Then when you look for 4th order decimal point accuracy, we have the high resolution mass specs, which are QTOF, quadruple with time of flight coupled. And then we have other high-end HRMS instruments that can give you resolution, sensitivity, at the same time solve your very low trace level detection issues, and then the quantitation. Software is our forte, and we have been doing very good in this platform in order to introduce newer softwares. Recent ones are ICH Q14 guidelines that says you should develop robust methods which can be transferred seamlessly to the quality control labs, and those methods must satisfy the FDA and ICH Q14 guidelines in terms of design of experiments, arrive at a design space, and be within the limit so that you are able to transfer those methods which do not fail at the QC. Chemistry, consumables, columns, various sizes, dimensions, chemistry, selectivity are again offered by Waters. Thermal analysis is one of our product line, which you may be familiar with TA instruments, DSC, TGA. These are the equipments which are used for rheology and polymer characterizations. These are the markets we address: pharmaceutical, pharmacy colleges, research institutes engaged in pharmacy like CDRI, ICER, and many other research institutes who are into pharmaceutical research. We always cater to their needs in terms of R&D as well as QC and data management. Then comes the material science, which is chemical material, food and environment. And India is highly populated, so we import food, we export food. Both are to be tested as per the regulatory guidelines of a specific country where you are exporting or when you import. So, someone said, what we eat that matters. Okay, so that's where the food is critical to today's scenario where there are a lot of regulations coming. Yes, research, which means I'm in biomedical research. All of you know how critical it is. The biomedical research, especially with the current COVID pandemic situation, and the clinical trials, the clinical applications, which includes diagnostic as well. Because the sample load is pretty high these days, whether it is RT-PCR or it is any other testing that has to be done in order to augment those doctors' prescriptions. Now coming to the Empower software, which I have highlighted in my keynote address, is basically Empower is one of our softwares that goes into the chromatography equipments laboratory. And you see here our Alliance HPLC. There are three things it does very nicely. One, it controls the equipment, it acquires the data, and then it processes the data. When I say it processes the data, I mean once you acquire the data, you need to process the data and you need to report the data. That is where it is very critical. There are certain integration processes which are to be used, automation process, thereby manual integrations can be avoided, and the data will be from system to system, chemist to chemist, analyst to analyst. It will be reproducible, and auditors will be satisfied when they integrate or when they rather locate your data in your system. And then comes the database. If you are on a workstation, single computer with a single instrument, there is also a database inside. So that's where we are from the beginning, a company which is Empower software chromatography product embedded on Oracle database. So that is why your data is secured. You don't lose the data. Even if there is a hard disk crash, we can retrieve the data. We can give it back to you. Nothing to worry. Now comes to scalability. You can start with a single computer, with a single HPLC. You can, as you go along and expand, you can have these additional workstation QSN software from Waters. And when you are an enterprise, like let's say Teva or let's say Indian company Sun Pharma or any other bigger companies, even the mid-sized companies these days are deploying the Empower Enterprise softwares where there are multiple GCs and multiple HPLCs. In addition to GC and HPLC, we also have the Empower servers that can go to many other technologies. I was explaining about UPC2, which is convergence chromatography based on supercritical fluid chromatography. I was explaining about EPLC, which is ultra performance LC, and APC, which is advanced polymer chromatography, which is the newer version of GPC, and capillary electrophoresis, dissolution. All these equipments from different vendors can be integrated into Empower platform. So the advantage is one software, your user is experienced to handle Empower software. He learns, he can handle almost any HPLC make and almost any GC make. Software remains the same. Your productivity of the laboratory increases, and you have a harmonization, and the data resides in the Empower server. It is easy to audit, and it gives you a lot of technical procedural controls There are, of course, every lab will have a SOP, standard operating procedure. They will have procedural controls with respect to their operations. But when it comes to technical controls, Empower ensure that everything is in a tighter regulated environment. These are our customers across India. These are the pharmaceutical companies who use our product. And I have not mentioned about the educational institutes here, because what I try to capture is the industries who have these references of Empower softwares, and there are obviously our MS, our LCs, our EPLCs on this platform. So with this short presentation, now I would request the convener of the meeting, Dr. Rupali, to take over. Thank you so much. Thank you everyone for participating.
Thank you, sir. Thank you so much. Kindly accept the certificate as a token of appreciation, sir. And thank you so much for your valuable guidance and your precious time.
Yeah, thank you so much. Thank you everyone.
Okay, sir. Now I declare that inaugural function is over. Let's start our technical session with the first resource person, Dr. Ritesh Oza, Senior Product Specialist, Mass Spectrometry at Waters India Private Limited, on the topic Use of QDA Mass Detector with CAMAG HPTLC MS Interface. Welcome, sir. Before we start with our technical session, I would like to give few instructions to the participants. As we are conducting this webinar on Google Meet, only first 100 participants will be admitted. Rest has to attend webinar streaming on our YouTube channel. YouTube link has been shared in the Telegram group. I request everyone to subscribe our channels. Subscribers' attendance will be considered for webinar. Please inform to your colleagues and your friends those who are willing to attend this webinar. Participants are informed that attendance link and feedback link will be shared in the chat box and in the Telegram group towards the end of technical session. Now I request Dr. Padmaja Kore, Assistant Professor, Modern College of Pharmacy, Nigdi, to continue with the technical session.
या आता चालेल.
Thank you, Rupali Madam. A very good afternoon to one and all. Myself, Dr. Padmaja Kore, Assistant Professor, PES Modern College of Pharmacy, Nigdi, welcome you all for the national webinar on QDA Mass Detector with CAMAG HPTLC MS Interface Technique video demonstration in association with Waters India Private Limited, Mumbai. It is truly stated that knowledge has no value unless you will put it into practice. So today we are gathered here for video demonstration session of HPTLC MS system. Without wasting much time, I would like to introduce today's speaker, Dr. Ritesh Oza, Senior Product Specialist, Mass Spectrometry at Waters India Private Limited. Speaking about his educational qualification, his PhD in chemistry, MSc in bioanalytical sciences, and BSc in biotechnology. Now, speaking about his professional experience, he has 12 years of experience in analytical instrumentation industry. Also, he is experienced in sample preparation to column chemistries to separation technologies in mass spectrometry. He is hands-on expertise on chromatography, spectroscopy, and mass spectrometry techniques. He has handled some technologies as automated extractors, automated nitrogen analyzers, SPE cartridges, LC and GC columns, HPLC, LSMS, LSMSMS, and HRMS systems. He has marketing experience in pharma, biopharma, food, feed,
environment, clinical, and biomedical research. He has also worked as an adjunct professor in biotechnology during PhD work. With this brief introduction, I would like to request Dr. Oja to start with the session. Over to you, sir.
Yeah. Thanks a lot for that introduction. And thank you, the college, for giving us this opportunity to discuss about our technologies to the young and the budding scientists, and also to the guides and the professors who are guiding them in their research projects and showing them the path forward. So thank you, everyone. So with this, I'm just sharing my screen. Just let me know if it's visible. Thanks a lot.
Yes, sir.
Yeah, is my presentation visible?
No, sir. Your screen
Hello?
is not yet visible.
Not yet, doctor.
Yes.
Not yet? Okay. Just give me. Yeah, is it visible now? Padmanabh, if you can just confirm.
Not yet.
No, sir, it is not yet visible.
No.
It's not yet visible. Okay. Okay, now is it visible?
Yeah.
Okay,
Yes,
great.
we can see.
Okay, thanks a lot, everyone. So, hi, I'm Dr. Ritesh, and I've been already introduced very nicely by ma'am right now. So, today we'll be talking about the use of Waters Equity QDA mass detector along with the ChemEg HPTLC interface. So, today we'll be talking about this innovation that we have established from Waters that we have developed in terms of a compact mass detector which goes by the name of Equity QDA. Now, if you look at the name QDA, Q stands for quadrupole, DA stands for Dalton, and hence that's how the name QDA came up. And then we have developed an interface like along with ChemEg, which helps in interfacing our mass detector to their TLC. So now people here would be aware about what TLC is, what HPTLC is, what is mass spectroscopy. Hence, what we'll be doing is, for the betterment of all the participants, we'll be first talking about the basics of all of these technologies, the basic principles of them, and then we'll look into what are the applications or the workflows where you can benefit with this innovation that we have developed. And then certainly at the end, we'll be open to answer any questions from the participants. So this would be the agenda for today. So we'll be first looking into what is the basics of mass spectrometry, means what are the basic principles on which mass spectrometry is based on. So what are the different components of mass spectrometry? What are the different technologies, or what are the innovations that have come up in these technologies? And when we talk about, say, the interface, or when we talk about the inlets, what are the different inlets which are available, and how TLC and HPTLC at times have their own advantages for this particular collaboration. Next, after going through the basics of mass spec, we'll look into more of the technology part as to what do we mean by the Equity QDA mass detector, what is in there for the researchers, for the young budding scientists who are at times so excited to know about technologies like mass detection. Because, like earlier, HPLCs were like a new thing for people working in colleges or in universities. Now, HPLCs are become really common. Now, the next trend which is moving ahead is for the mass detectors. As to what more can you do with MS, how can you handle MS, and what are the type of expertise or the skills that you need when you enter any industry. This is important from the perspective of the students as well, because here we have students who are doing B Farm or M Farm, and they are the people who would be going into the industry and working on these equipments. And hence, if from the initial days itself, if they have an understanding of what is going on in the market, what is there in the market now for some of the pharmaceutical challenges, then this acquaintance will really help them in the long run. After having spoken about the technology, we'll just go through what are the workflows. For now, we'll focus on the natural product analysis workflow. What are the different steps in natural product analysis, which includes, say, extraction, purification, then final analysis. And then we'll look into what are the different challenges that you might encounter as scientists, and how we have developed solutions which will help you to get away from those challenges and become more effective and more productive. Finally, we'll come down to QDA with the TLC-MS interface as to what is this whole technology about. Because since years, people have been talking about GC and MS. People have been talking about LC's interface with MS. But then with this TLC-MS, we have coupled one of the most regularly used technology or a technique in labs, like in the research labs and pharmaceutical labs, in universities, which is mainly the HPTLC, and how it has been interfaced to a sophisticated technology like mass spectrometry, and how does it open a gamut of applications to which you can use this combination of these technologies. So to begin with, we'll just go into what are the basics of mass spectrometry. So what is a mass spectrometer? If we go into the definition, a mass spectrometer is an instrument that measures the masses of molecules that have been converted to ions. That is, molecules which are electrically charged. Now, this fundamental definition itself is very important because it defines somewhere the process, what is happening in a mass spectrometer. So a mass spectrometer, it measures the masses of the molecules. That is first. How those molecules are first converted into ions. And what are ions? Ions are nothing but molecules which are electrically charged. So this introduces us somewhere to the steps in a mass analysis, wherein it talks about ionization of the molecule, wherein first you have to ionize your molecule for them to get detected by the mass spec. We are therefore measuring the mass to charge ratio of the analyte of interest. A word of caution here, that here we are not talking only about measuring the mass, but we measure the mass to charge ratio. This is a very important fundamental point which is to be remembered when we talk about any mass spectrometer. So here, first we ionize the molecule so that they get charged. Once they take up the charge, they are measured or they are separated on the basis of their mass to charge ratio. If you compare it with the colloquial terminologies, so basically what you're trying to do is you're trying to measure the molecule. Means this is just equivalent to a weighing scale, for example, wherein you weigh a couple of grams of certain compounds. So with a similar analogy, you're going down and you're trying to weigh the molecules themselves. So this is the whole idea about the mass spectrometers. But now an important point to understand here is that the mass is a fundamental property of any of the molecules. And hence, if you want a universal technique in terms of the detection or the analysis of certain molecules, then mass spec is the only option which is available. Because some compounds may have some chromophoric groups, may not have some chromophoric groups. They might absorb the UV light, some of them might not. So hence, for such compounds which say are chromophoric or non-chromophoric compounds, irrespective of that, you can do the analysis depending on using the mass spec detector. So with this fundamental in mind, now let us try to understand what a mass spec is all about. A mass spec first is considered having the sample introduction system. Now, what is the sample introduction system? A sample introduction system is something which will introduce your sample along with the analytes of interest in it into the mass spec. So if you have compounds which can be dissolved or which are miscible and can be separated on an LC, so you separate them on an LC using an LC column, and those elements are passed into the mass spec. So that is called as an LCMS analysis. If you are working on compounds which are volatile, then you use GC columns, you separate them using a GC, and then you do the GCMS analysis. Now here we have people who are doing pharmacies, so you might be aware that there are solvents, there are volatile solvent analysis that is a part of the routine pharma analysis. So for solvent analysis, you consider this is an example wherein you have a GC which is coupled to an MS for GCMS analysis. You can have an headspace which is connected to the GC, so you have headspace GCMS analysis. So today what we'll be talking about is we'll have one more dimension to LC and GC, which is the TLC, which is the thin layer chromatography, which can also be used as a technique of sample introduction. So basically you separate your analytes of interest depending on certain properties in LC, GC or TLC. Once you have those separated compounds, you try to identify what those compounds are, or you try to quantitate how much of those compounds are there in your sample. So once you have your samples introduced, then this is the ion source. Ion source is the area where ionization happens. This is where your molecules which are uncharged, they get the charge. Here, depending upon the type of inlets that you have different type of ionizations which happen. Like for example, for GC, it is mainly electron impact, or you have the chemical ionization. In case of LCs, you have the API ionization, which is nothing but atmospheric pressure ionization. So there again, you can have electrospray ionization, that is for polar compounds, or you can have APCI, which is atmospheric pressure chemical ionization, which is for mid-polar to non-polar compounds, or you can have atmospheric pressure photoionization. Once you have your molecules in their ionized form, which is now they have the mass to charge ratio, you can separate them on this basis using the mass analyzer. Now, in terms of the mass analyzers, for the information of the participants, there are different technologies that are now there in the market. So this topic itself is an extensive topic of research and of interest these days, because there are many innovations which are happening here. Like in mass analyzers, there are quadrupoles. Then in quadrupoles also you have single quadrupole and triple quadrupole. And then you have time of flight instruments. In time of flight, again, you have hybrid instruments. Means you have a time of flight coupled to a quadrupole, so that's a QTOF. And there are multi-reflectance systems also, wherein there are multiple TOF tubes. So with the mass analyzer, which is basically the heart of an MS system, because this is where you are separating your compounds actually in the system. Then once you have separated your ions, those they go into the detector, wherein they are converted into a signal, and then you get it in as a data. So then you have your softwares which are there in your PCs installed, which acquire and help you process the data. So this is what a mass spectrometer is about. It has ion source, it has analyzer, and it has a detector, followed by a software which will do the acquisition and the processing. And finally, once you have this in place, you finally have the results. So this can be your qualitative analysis results, or this can be your quantitative analysis results, or this can be both of them. So now let us try to understand the difference between a TLC and an HPTLC. Looking at the terminology itself, that we understand that TLC stands for thin layer liquid chromatography and HPTLC stands for high performance TLC. So like we see the difference between the two, here what we see is this is a traditional TLC plate in which the samples have been manually spotted and they have been run through a solvent chamber. Whereas here we see that there is no that great separations, because here we see many of the compounds coming close to each other. So this is the standard and these are your samples. Now with the innovations which have happened in the type of silica which is being used with the automated impregnation of the silica onto the plates, so hence we have these high performance TLC plates which are now available in the market. And with the innovation into loading the sample, we have automatic sample loader. So you see that we have a very fine band of samples that can be loaded and they then can be separated. So there what we see is that here we see much better separation among the. So these are your standards, and then parallelly you can check your sample to your standard ratio and you can do the quantitative analysis also. So then in this world of LCs or liquid chromatography or gas chromatography, which are sequential analysis, wherein you have to analyze one sample after the another one, what are the advantages of a planar chromatography? There is nearly unlimited choice of stationary and mobile phases, because with innovations there have been many different chemistries of stationary phases that have been developed, so there is no limit to it. And very important example is, advantage here is multiple samples can be analyzed in parallel. Means you can have 5, 10, 12 samples which are getting analyzed at the same time. So the idea is that your standards as well as the samples, they are getting exposed or they're getting run in exactly the same condition, exactly the same temperature conditions, environment conditions, having run in the same solvent composition at the same time. There is single use stationary phase, so there is no cleanup of the phase which is required, wherein you have to clean or you have to wash the plates and you have to use them again. So this is a single use stationary phase. Archive of separated substances on each track, so you can do multiple detection on the same plate. That is possible. So if you have compounds which have different properties, then you can do detection of multiple different properties on the same plate. You can do quantitations also, means you can run your standards like that has been shown here, and you can compare it with your samples, and then you can do the quantitation studies as well. Nowadays, TLCs have been used for not just the chemical detection, but also for biological detections. Directed and cost-effective mass spectrometers are also available. And hence, all in all, what you can see is, you can actually see the separation when it comes to a TLC. Whereas when we talk about LCs or GCs, you have to rely on the data which is coming out in the software in form of peaks. Whereas in TLC, you can see the separations or the bands directly on the plate. It is reproducible, because if you have the same environmental conditions and the same solvent conditions being maintained, you are guaranteed of the reproducible performance. And if we compare it with the hypher-tended techniques, this is really cost effective, considering the cost of the TLC plates, the HPTLC plates that they cost, and overall time of run which it takes for the plates to run to the 3/4. This becomes a very cost effective method compared to the other methods which are in the market. So with this, you can develop something known as an HPTLC fingerprint. This terminology is really the trend these days when it comes to natural products, because natural products, we know that only a particular section of a plant, if it is having certain characteristics or properties, then only that has to be used. And every particular section of the plant would have a characteristic HPTLC fingerprint. And hence, when we talk about phenomena like adulteration happening, you can detect those adulterations using the HPTLC fingerprint very easily. So you can do the comparison of tracks from different plates and analysis file. It's a useful tool for identification and detection of adulteration in the quality control of raw materials. Because if the raw material itself which is being used, it's not of the correct plant part, or if it's been adulterated with any other plant or any other plant part, then in that case this becomes a very grave challenge. So then now let us understand. So now we have talked about the sample introduction part. We have talked about the TLC and the HPTLC. Now let us talk about the electrospray ionization. So this is, like the term says, ionization. This is the first part, like we just discussed about a mass spec, where the ionization happens. So now what happens here is the sample is introduced through a narrow stainless steel fused silica capillary, which is having an ID of 50 to 100 microns with an appropriate flow rate. A voltage is applied to the tip of the probe. As a result, a high electric field, the sample emerges from the tip as an aerosol of highly charged droplets. The nebulizing gas helps with directing the spray towards the analyzer region. So what happens is that, consider this as your eluents which are coming out from your sample introduction system. So it is coming at a very high flow rate. Once it is coming in, this is surrounded by a concentric tubes which pass a gas, which is nitrogen gas, which is at a temperature of 500 to 600 degrees. So now what happens is, because of this high temperature, whatever eluent which is coming out, the solvent gets evaporated. Now as the solvent is getting evaporated, what happens is you get desolvation which happens. Secondly, what we see is that as this is coming at a very high flow rate from a very narrow lane, what we see is that there is a spray which is getting generated. Now because of this spray, what happens is the overall surface area of the droplets is increased, and hence because of this, what happens is that as it moves away from this inlet, the solvents get evaporized, and hence the droplet size goes on reducing. As the droplet size diminish in size due to the solvent evaporation, assisted by the drying gas, eventually charged sample ions free from the solvents are created, and some of them will pass through the lenses into the high vacuum system. So at the end, what we see is that here, as the droplet size will go on reducing, the charges, like we know, like repel, the charge physics which is there in place, and hence because of this, what happens is that there is a Coulombic explosion which happens, means like repels like, and hence because of this, what we see is that we see individual ions being coming out from the droplet. In this process, as they move away from the inlet to further into the source, what we see is the ions which are generated, they enter this high vacuum region. Once they enter the high vacuum region, now what we see is the shape of this cone. It allows only the ions coming into the system, and hence your uncharged molecules and your droplets are not able to enter, because here again we apply electrical voltage, and because of this electrical voltage only your ions are able to enter in. Next, what we have to see is that here we are maintaining a pressure of 1 millibar, whereas when it goes into the mass analyzer, the vacuum is around 10⁻⁻⁴. So, and hence your analysis further happens in vacuum. So just confirm if this the sound of this video is visible. Sorry. Were you able to hear the sound?
Yes, sir.
Yeah. Okay,
Yes, sir. It is clearly audible, sir.
Okay, thank you.
No, sir. There is no voice in the video.
No voice?
No voice. Yeah.
No, sir.
Okay. Yeah, just give me some. Okay, fine then. Okay, so then let us talk about the quadrupole analyzers. Now, after the ions being generated in the source, what happens? Yeah, yes.
Sarji, if you unmute yourself during the video, then voice will come.
I've already means if I mute myself, that's what you mean, sir?
No, you have to unmute yourself. Unmute during the video.
Okay, let me just try.
check sir, check. म्यूट और अनम्यूट करके देखिए, सेटिंग थोड़ा तो आवाज आ जाता है. Now play the video.
Yeah. Yes, sir.
नहीं सर, नहीं अभी भी नहीं आ रहा. आप अभी अनम्यूट करो, आपको अनम्यूट करो.
अनम्यूट ही है सर मेरा. आई एम अनम्यूटेड ओनली.
म्यूट करो, आपको से म्यूट करो.
So try once, you mute yourself and try once, you can see. No, sir, it is not yet audible.
Okay.
Can I suggest you, instead of making a window share, you make a tab share with sound click. Okay, so when you are doing sharing, so instead of sharing window, you share tab and the sound when you are sharing the tab, then the sound will be shared.
Okay. Let me do it this way.
Just take it.
Okay. Is it audible now?
No.
Mute yourself. No, you have to stop video and you mute yourself because the echo is due to the your mic and the sound of your video.
Speaker. Okay, get it.
You have to mute yourself and then you have to share. Ritesh sir,
Yeah.
you have to share all your screen. Only the tab you have to share where you are playing the video. Okay, so you start video in one tab of your
Okay.
window and share that window only, and mute yourself and other things so that it will not make any echo.
But then when I would have to talk after the video, then I would have
Yeah,
to
yeah.
again.
Then you have to unmute yourself.
So then that in that case, I would have to minimize the video again, and then I would to do it, right?
Yeah. You have to stop
So
video and then you have to.
so what I will do is for now, just for the continuation of the flow, we'll skip on the videos, and I'll
Yeah.
share this presentation with everyone with the videos in them, so everyone can then refer to the same.
Yes, sir. Sir, Ritesh sir, meanwhile,
Yes.
you can WhatsApp me the video. I will share from my side.
Sure, sir.
Possible.
Sure, sir. I'll do it. Yeah. Thanks. So now, am I audible to everyone?
Yes, sir. Yes, you are audible, sir.
Okay, great. Okay, so after once you have your ions generated, now the next step is that how do you separate those ions? So in the next step, which is the mass analyzers, your ions, they are separated.
Sorry for the interruption, sir. Your slide is not visible.
The slide is not visible.
Presentation is not visible.
Okay. Now is it visible? Can you please confirm, ma'am?
Yes, sir. Yes, is visible, sir.
Okay, great. Okay. So now what happens in the next step is after your molecules are ionized, so these are your ionized molecules. Now they have to pass through a set of quadrupoles. Now what do we mean by quadrupole is, quadra stands for 4. So we have a set of 4 poles through which the ions have to now pass through, and we apply an RFDC voltage. So this RFDC voltage will help to separate these ions on the basis of their mass to charge. So this is how the movement happens, that ions which are not of the target mass to charge will hit the rods and they will get rejected, whereas your target mass to charge, which is dependent on the RFDC voltage, only that particular ion will pass through and will enter the mass analyzer. And in that also, only the ions which have the specific mass to charge within the range, if you're setting a range of say a lower and a higher range, then only ions within that range will pass through the quadrupoles, and they will get separated on the basis of their mass to charge ratio. So this is how the separation of masses happen in case of the quadrupole analyzers. So now we have talked about the analyzers. After the analyzers come the detector. In case of detection, this is where your ions, they generate the signal and your software picks up them and gives you the final result. Next, what we'll do is we'll just go through the equity QDA, which is a compact mass detector from Waters. So what does this help in? So this is designed for chromatographers. So this is the whole idea as to why we developed this mass detector. Is this was for the use or for the betterment of the chromatography people who are facing issues about detections of certain of the analytes of interest. So this is what Dr. Devi from School of Pharmaceutical Sciences at University of Geneva had to say, that the equity QDA has been specifically designed for chromatographers. Here it is important that here the idea is that even people who are working on an HPLC or an LC system can use a mass detector. It does not require very special expertise that only people who know the mass spec technology or some mass spec software can work on this instrument. Because the idea here is that the controlling software is the same. for the LC as well as for the mass spec. And both of it is the Empower software. So Empower is the software that my colleague Mr. Prakash Mohanty explained in his talk. So using the same Empower 3 software, you can use the HPLC as well as you can connect this mass spec detector. And hence, this is designed specifically for chromatographers. It is as easy to use as an optical detector. So it is just like using a UV and a PDA detector, and does not necessitate specific training or expertise. The detector is well adapted to speed up the method development process through efficient peak tracking and minimizes the risk of co-elution. So here you can achieve two important advantages. First is the peak tracking and another is the risk of co-elution. So what these two challenges are, we'll discuss about them in the next slides. So what happens is, in a traditional LC separation, now if you consider this as your LC separation of 1, 2, 3 and say 4 peak, looking at this chromatogram you will see that okay, you have got 4 well resolved peak from the LC. But if you try to dig in more and if you have your data in 3 dimensions, that is the retention time here, then the wavelength in this particular axis and the absorbance which is being observed, then you will get some interesting information. What is that? That first, there is an co-elution also which is happening, that this particular peak number 3 is actually having 2 masses in the same, and hence though it is appearing as a single peak, it actually has 2 molecules, one having molecular weight 284, another one having molecular weight 306. Also, there is one particular peak which is of a non-chromophoric analyte. So that is also having a molecular weight of 304. So here you are missing one particular peak. So if you rely only on a UV or a PDA detector, which rely on your compounds having the chromophoric properties, you would miss them. Sorry. And hence here you can detect non-chromophoric compounds also. So these are the two important advantages with a mass detector. And why it is important is because in natural products, many of the compounds, many of the metabolites which are there, which is the secondary metabolites from plants, they are non-chromophoric in nature. And hence, this is where a mass detector actually helps. So how do you move upwards from an optical detection? So first, if you consider a UV detector will give you low selectivity and it is more for routine or targeted analytes. So you know that, okay, this is the lambda max of your compounds. So you can use the TUV for those specific analytes. And hence you know that, okay, this is what I am looking at, I want to quantitate, then you can use a regular UV detector. When it comes to selectivity, with the PDA, you get more selectivity, and this will help you to make some decisions, wherein you have to check for peak purity analysis. This is where the PDA detector will be more useful. Now, when you move ahead from a PDA to a QDA detector, what happens is you get very high selectivity. So you can set that, okay, this is the molecular weight of the compound, I want to concentrate only on this particular mass. Then you become very selective. You give method development certainty, means because of the peak tracking advantage, you develop much more confident methods. Expanded range of analytes. So like we saw in the previous slide, you can not just focus on your chromophoric compounds, but also the non-chromophoric compounds. So this expands the range of applications that you work on. Improved profiling in matrix. You get better speed and you get better sensitivity. Because here, in terms of the sensitivity, it is better than a UV or a PDA mass detector. And hence, if you are say working on applications like genotoxic impurities, then in that case, this is a very big advantage. So when we look into the confidence level increases from a UV to a PDA and a QDA. When we talk about confidence, this is about the confidence in the decision making which you can get while using a QDA mass detector. So what are the advantages? This is a detector concept. So this is just of a size of a UV or a PDA mass detector. It seamlessly integrates into any water system. So here what we see is that if you have a prep LC, so if you are doing some synthetic chemistry reactions and you want to purify your compounds, or if you are working on some natural products and you want to do a prep LC purification, along with the fraction collection which is there, is not dependent on a signal from an optical detector but on the molecular weight. So with this you get better fraction collection, which is much more accurate. You can connect an analytical HPLC from water, you can connect an UPLC from us, you can connect a UPC square, which is the supercritical fluid chromatography from us, and you can also connect a UHPLC. So depending upon the type of project that you are working on, say for example at present, if you want to connect it to an HPLC, you can connect it to an HPLC. So students, then if you go into the industry, HPLC has now become a bit routine. So people will then expect you to know what is UHPLC. So if you're using a UHPLC there, then students, you can connect a QDA to a UHPLC also. And now with the trend moving forward, people are now looking into UPLC more. So there you can connect a QDA also to a UPLC system. With a lot more focus being going into chiral impurities and separations of chiral compounds, many companies are now looking at chiral separations using the SFC system, which is the UPC square, because it helps in two advantages. First, it reduces the solvent consumption, and at the same time your process becomes more environmental friendly because you are using the carbon dioxide under high pressure as the mobile phase here. So this is how the QDA can help you in whichever type of separations that you want to do. You can use the QDA. With this, you can work in the single ion monitoring, or you can work in the full scan mode. Means if you want to do targeted quantitation, then you can do it with the QDA. So this will give you better sensitivity. Or if you want to do some profiling studies wherein you want to see the molecular weight of all the compounds which are getting eluted out, then you can do a full scan analysis also. Important point here is that the calibration of the mass spec happens automatically and it is invisible because the calibrant itself is a part of the system itself, and hence the system automatically calibrates itself the moment you start it. So if you are just switching on the system, then within 7 to 8 minutes you are ready to go. Also, this can be controlled by our Empower software. So using the same software, you can control the LC system as well as the mass detector. And then we have a dedicated MS software also, which goes by the name MassLinks. So you can control this by using the MassLinks also. This has a mass range of 30 to 1250. So this includes most of your small molecule applications, whether it is natural products or it is synthetic molecules. It has pre-optimized electrospray ionization in both the positive as well as in the negative mode. And with this you can do quantitations also. So because of which you can attain around 4 orders of linear dynamic range. So how does this technology actually looks like? So like we discussed earlier, this is the source, this is your mass analyzer, and this is the detector. So here you have your ESI capillary. So this is where your eluent from the LC is coming in, or for example, today we'll be talking about TLC. So your TLC's separated band should be coming in through the system. Here, this is the place where you apply a temperature of 600 degrees and you apply a voltage of 0.5 kilovolts. So because of which you get the desolvation and your molecules get charged. So once you get charged molecules, they enter into the sample cone. Once they are into the sample cone, they pass through this ion guide. So this ion guide then focuses your ions into the quadrupole. Now, important point to know here is that we see that this particular yellow shape is actually the path of the ions which they take from the ionization source to the quadrupole. Here we see that the path is going through two bends. So students, this particular bend is actually helping only the ions to get into the system, and your neutrals are getting removed. So the advantage for you is that if you are using any buffers, or if you are working on any dirty matrices, like from your synthetic chemistry samples or your natural product samples, then your matrix or uncharged molecules will not enter the mass spec. Once they enter, your ions are into the ion guide. There also what we see is that the ion guide, it does two functions. It is actually just passing your ions from the source into the quadrupole, where they would get separated. In the process, what you see is that this is going through an S-shaped path. Now this S-shaped path is nothing but a path which again your ions will take, wherein they will move from the lower channel to the upper channel. Also, that you can observe is that this lower channel is much more broader, whereas the upper channel is very narrow. So because of which your ion focusing is much better, and hence you are not losing any signal, hence moving ahead, and hence you can do very high sensitive analysis also with the same instrument. Further, what we also see is that the neutrals and the uncharged molecules further do not enter the instrument in the ion guide. So these two innovations, which is the Z-spray source and the step wave ion guide, they improve the productivity of your lab. So this is important, friends, because today you are working on research projects, but tomorrow if you are working in the industry, then the industry, you need to be aware about technologies which can help you to become productive from the first day itself. And hence having technical awareness about such features will definitely help you in the long run. Like we mentioned, there is no tuning required because there is an inbuilt calibration which is there in the system, and this source is optimized for both UPLC as well as for the UHPLC separation. Next, what are the applications that you can use a QDA for? So whether you are working, say, for a research project for method development, wherein you are trying to optimize an existing method, then you can use the QDA there. If you want to do some sample profiling, so then you can do the sample profiling also. Like, because here what we see is that here we are analyzing vitamin D3 as well as glucosamine in this sample. But in an optical detector, we are able to see only vitamin D3 and not glucosamine. So hence with the glucosamine, analysis can be done using the QDA mass detector. So you can do sample profiling also. If you are working on synthetic chemistry, this is something which is booming these days, because as there are new diseases coming up, people are trying to innovate more. They are trying to find new drugs. And if you're doing some synthetic chemistry applications, then to monitor whether you have got your products being formed, and at what level or at what time, under what conditions, then you can do your synthetic chemistry applications also with this QDA mass detector. Because in this, what will happen is, as your reaction progresses, you will see that the mass or the response for the products will go up, whereas the response for the reactants will go down. So hence with this mass detector, you can do synthetic chemistry analysis also. Next, if you're working for some purification work, like for some natural products or for some impurity isolations, then those can also be done using the QDA. So next, what are the benefits with this system is that it helps to streamline the workflow and also improve the lab efficiency. Now it also happens to give you low return on investments. Why? Because, the running cost is low because the power consumption is less. The service plans, because the maintenance is really very simple. There are a couple of parts that are to be changed so that the user can change themselves, which includes the sample cone and the capillary. You can simply turn off after use, because once your work is done and you don't want to use it, you can just switch it off. If you compare this to any LC-MS instrument, those have to be kept on for 24 hours because there the vacuum has to be running continuously. At the same time, it is space saving, because like we see in this picture here, this is your LC rack. You can just place your QDA above the same rack, so you do not need any additional space. Standardized on QDA and achieve substantial saving over typical ownership duration. This is how you can save. This is also useful for colleges, because here you won't have no very high experts who will be working on exposed. The students have to learn the technologies, how to go about using them, and hence it is very good and easy to use. At the same time, there is no high running cost in terms of the solvent consumptions, and also the maintenance cost is lower. So colleges and universities can look at this system as an orthogonal detection technology. Next, what we see is when we talk about method development. In method development, when we try to change the pH, that is from low to high pH, we see that there are different conditions that happen. Because what happens is, when you have your mass data which is linked to the UV chromatogram, what you see is that, okay, this 258 molecular weight which is there, it is coming at the same retention time. But because of this change in pH, what has happened is that other peaks have shifted. Means you see that this particular 196 which is there, which was earlier coming at 2 minutes, is now coming, it is coming at less than 1 minute. Whereas another peak, say this 202, which was also coming at around 2 minutes, now it is coming within 1 minute type of analysis. Then then you have 300 and 342, which are coming very close here. But when you change the pH, they are getting separated within around 2.7 to 2.8 minutes. So this is the benefit of the mass detection, because you can actually, from the molecular weights, you can understand that, okay, how the peaks are getting shifted, or you can do the peak tracking. So this is what is the benefit of peak tracking feature in a QDA mass detector. Imagine that if you wouldn't have this mass detector, then you would have to run your standards as well as samples in both of these conditions, compare them, and then try to see that, okay, how your peaks have shifted. But with this mass detector, you do not have to rerun. You just have to run them in the low pH. You have to optimize your method. You have to do some separations. You take it to high pH, and you, because of this peak tracking advantage, you can track how your peaks are moving around in the chromatogram. So new assigned mass field automatically assigns masses, uses base peak for peaks of the same mass but different retention times. So means if you see that, okay, the same peak is coming at two different retention time, means there is a peak splitting which has happened, and the peak tracking reporting table of retention time summarized by assigned masses. Each mass present in the sample by retention time. So you can check that, okay, which is the retention time, what is the RT, and what is the assigned masses. So this is the type of peak tracking application that can be done using this. So you can see that, okay, this is your CSH column, this is the mobile phase composition, and what is the retention times that you have seen, what you have observed. Next, you might have a condition wherein you say that, okay, my peak looked pure, but the core MS lab found otherwise. Means here you might see that, okay, this is just one single peak. But when you send it for mass spec analysis, the mass spec analysts say that, no, this peak is not pure. And how do they say so? Because if they compare the leading part of the peak, they will see that there is a mass 300. When they touch upon the apex, you will see that there is actually a mass 300 and 342. And at the trailing, there is 342 again. Hence, this ensures, further reconfirms that this is not a peak, a pure peak. So with this, you can check peak purity using both mass and PDA detector. So the idea is that you can connect a UPLC So the idea is like we've shown here, you can connect a UV followed by the QDA, or you can have a PDA which is followed by a QDA mass detector. So the advantage is in one single window, you will get the UV chromatogram. Followed by that, you will also get the mass spec information. So you will get the UV spectra, like this is having a lambda max of say around 261 somewhere around, whereas the molecular weight is 302. This one is your second peak. This is having molecular weight 302. The third one is having molecular weight 314. So likewise, when you couple the optical detector followed by a mass detector, you can get much more comprehensive information about the same sample. Along with this QDA, now we said that, okay, there is a desolvation gas also which is required, or you need nitrogen there. So now people can get this nitrogen from a nitrogen generator. And with the QDA, we have a compact nitrogen generator also, which supplies around 35 liters of high purity nitrogen in laboratories that utilize an external source of compressed air. So the compressed air source is required, as well as being stackable within the specific equity QDA system configuration. The generator is also wall mountable. So you can stack up this nitrogen generator along with your QDA, or you can just mount it on to the wall. So this is important for labs wherein there are space constraints, and hence this becomes a very good space saving design as well. In terms of LCs from Waters, we have the HPLC, the UHPLC, and the UPLC. So this is a very important slide for the students as well as for the faculty members, because this shows the trend which is happening in the LC separations today, and what are the key distinctive features of these technologies. Like if you are working with an HPLC, you are working with pressure limits of around 5,000 to 6,000, wherein the columns are 4.6 mm and the pore size is around 3.5 microns. Whereas in case of when you go to a UHPLC, the pressure limits are 9,500 to 10,000, wherein you use 3 mm column and the particle size is around 2.5 micron. Now the trend is now moving towards fast separations when people are using 2.1 mm column and 1.7 micron particle size. The QDA, like we have shown here, can be connected to any of these platforms. So where do each of these fit into? So if you are looking into monitoring or you're looking into routine QC analysis, then this is where an HPLC is used. If you're looking at characterization, monitoring, and plant transfer or QC analysis, again, this is where a UHPLC is used. Again, UPLC, many of the methods in the pharmacopoeias have been modernized, wherein the UPLCs are now being used for routine analysis as well. So the idea here is that the QDA can be used with each of these LC platforms. So what do the customers have to say about QDA is that companies that don't have mass spec super users on staff finally have a way to access quality mass data. People then also say that the QDA mass technology can be applied to support the whole life cycle of a drug from early development to manufacturing control. Because with this, your entire decision making becomes much more confident. This is a mass detector that we have been waiting for. This is a small and easy to use mass detector. Next, it's great to have power of mass data in our lab without the use of headaches or need of a mass spectrometrist. If you want a reliable, robust, small and easy to use MS, the QDA is your perfect choice. So this is coming from universities. So this is what people in universities or colleges or institutes feel, that they need a reliable and a robust system, which is not just simple, but it is also easy to use. And the QDA is the perfect example and the perfect answer to them. When we talk about maintenance, so like we have shown here that we have done around 1200 injections of plasma samples for this particular compound, Veramapine. What we see is that there are two parts which have to be changed. So first is the sample cone that can be changed easily by the user themselves, and the ESI probe capillary, so that can also be changed by the user themselves. If you talk about how easy it is, so you just have to switch on and it is ready to use in 10 minutes. There is an integrated calibration can be automatically performed upon startup. So you just have to switch on and the system has an inbuilt calibrant. So using it, the calibration is done automatically. So this is the ESI capillary tube. So this is just one consumable which is to be used. This is a single probe ESI. Any user can quickly plumb directly into the ion source and the outlet of the LC system. It has a pre-optimized geometry, so there is no adjustment which is required. So you just have to sort of unscrew it from the LC outlet and the MS inlet, and you just have to do it. That's it. It's a tool-free replacement. Even the students can do themselves. It increases the uptime because then you do not have to wait for any service engineer to come down into your lab and do that replacement. And it also reduces reanalysis, because if you identify that okay, the capillary has gone bad, you can just replace it and you can continue with the analysis. Like we said, the calibration is completely inbuilt and hence this happens automatically. This can be configured to check calibration upon power on. Inbuilt data confidence at the push of the button. So because of this inbuilt calibration, you can see that okay, the confidence in the results is also much higher. Now this is a case study. Now we'll be talking about pharmaceutical case studies because today we are talking to the pharmacy researchers and to pharmacy students. So this is an example of metachloramide. This is an anti-emetic used in treatment of nausea and vomiting. This is the main product which has a molecular weight of 219. Along with that, there are other impurities also that have to be analyzed, and these are their masses. Yeah. Any questions? Okay, thank you. So what we see is that using the QDA mass detector, you can distinguish between 300 and 316, because what would happen is that looking at the chromatogram, you would feel that okay, there is no great separation because here it is at 1.4 and this one is at 1.59. So the purity check will show them as passed, that okay, it's passed, but actually there are two masses which are co-eluting, and hence when you have a UV which is followed by a mass spec, you will see the molecular weight information also. So like these are the closely eluting peaks here. You can get the molecular weight information and you can easily judge that where there is a coelution which is actually happening. So what happens is, when it comes to regulations, new ANVISA requirement of degradation stability studies. What was the challenge there? Similar UV for two co-eluting peak. So that is at 1.423 and 5.425. 23 impurities, including non-chromophoric compounds and coelutions of analytes with the similar UV spectra. So here there are two challenges. First, you cannot see some compounds in UV because they are non-chromophoric. Second, in the ones that you can see, there is a coelution of analytes with UV spectra. This is where MS or the QDA helps. And hence there is time. So because of which, if you are not using a QDA, there is time consuming method development you have to use using individual standards, and it impacts the cost. Because when we are working as students, when you are working say in research institutes as students, we do not consider the cost factor. Or if we are working as a scientist, we do not look into the cost factor. But students, my friends, when you try to go up to the next level, you also need to look into the cost factor, because the research guides or your managers in the future pharma companies would be looking into the cost factor also. And hence, whenever you suggest another set of experiments, that would include a lot of cost in terms of the solvents, in terms of the consumables, in terms of using the same instrument again and again. And hence the cost implications are always on the higher side if you're not using a QDA. So with this QDA mass detector, what happens is you can detect the non-UV impurities detected by the QDA, and the QDA spectra highlighted the co-eluting peak at 5.4 minutes. So you can see that, okay, these are the two peaks and they have the two different masses. Method development speed is increased because the confidence is higher, and the confidence and improved quality of information can be submitted to NVISA. So when we talk about regulations, this is where it helps. When we talk about genotoxic impurities, these are the guidelines which are followed worldwide: EMEA, US FDA, and ICH M7. This is about a pharmaceutical company in India. So they had challenged in sensitivity of for impurity analysis. They were just using an HPLC with a UV. Now in that, they had to inject around 100 mg per ml of API concentration to achieve the adequate sensitivity. Now when you are injecting such a high concentration of the API, you are loading it onto the column, so your column life goes down. As the amount of sample itself is on the higher side, you get distorted peak shape, and because of the same high API concentration, there are solubility challenges also. So what are the challenge that you would face in such situation is method support, user training, and also the data integrity part. With the QDA, what happens is you can meet the regulatory requirement. The UV methods can be replaced using a QDA mass detector. You can consolidate multiple methods on the single platform. Improved column lifetime, because with the QDA, like we said, you can achieve higher sensitivity. So you do not have to load that much higher amount of sample. And hence your column life also increases, and hence your running cost also goes down. There is less delay through fewer solubility challenges. Why? Because you would have to use less amount of API, and hence the solubility challenges are minimized. So this is about the the QDA. So you improve your lab efficiency, as well as you get these indirect benefits as well. Now with the QDA mass detector, this can be coupled simultaneously to a TLC as well as to a UPLC. This is important for synthetic chemistry workflow. So till now we were talking about QDA with a UPLC. Now slowly we'll be migrating into QDA with a TLC. So with this setup that you're seeing on the screen, you can connect a QDA to a UPLC and parallelly to a TLC MS interface also. So whenever you want to do an LC based separation, you can use this diverter valve and you can allow the flow from the UPLC going into the QDA. So you can use the QDA for an LCMS analysis. Next, if you have some TLCMS, then what you do is you stop this particular flow from the LC and you allow the TLCMS interface to be getting connected to the QDA. So now your samples from the TLC will get into the QDA. So you can use this as and when required with an LC, or you can use it with a TLC. So then what is there in place with the TLC-MS interface from CAMAG and the QDA mass detector? So what are the applications for QDA with TLC is for academia chemistry and mass spectrometry teaching labs. So if you have some sophisticated instrumentation facility or any course wherein you are training the students on sophisticated technologies, this is where it can be used. Because if you're working on organic chemistry or you're synthesizing some molecules, you can do simple rapid reaction monitoring. It is affordable. It is a detector level ease of use. It is giving you unrivaled robustness and uptime, and it is easily transportable. Means you can shift it from one place to another without an issue. Complemented by direct loop injection via a divert valve. So using this divert valve along with the TLC interface, this can give you complementary advantages. So what are the highlights? QDA adds on existing CAMAG HPTLC installations. So if you have an HPTLC from CAMAG, you can take QDA as an add-on. So you do not have to purchase the whole set as together. So you can just add on a QDA to an HPTLC from CAMAG. So CAMAG are the #1 provider for an HPTLC equipment. So they are, whereas they're being used in cosmetics, natural products, and food. And hence, with this connection of QDA with TLC, you get mass data gets accessible for the non-MS experts as well. When we talk about another industry like cosmetics, major cosmetic industries have the phytochemistry department. HPTLC is popular in Europe, but in India, with many of the companies like Patanjali and many of the other companies like Himalaya, Dabur, focusing on natural products and people becoming more aware about the benefits, this is gaining importance. QDA provides the most accessible mass confirmation to add confidence in fingerprinting of botanicals. So like we discussed in the initial part, so wherein if you want to fingerprint your botanicals with respect to the plant and the plant part, with respect to the properties that they connect, you can do a fingerprinting using a QDA and you can establish a fingerprint using this mass detector. So here we see that, okay, this is the band that is what we are targeting, and we see that this particular band has this molecular weight for all of these. So if this is a standard and these are your samples, then you can be sure that, okay, it is the same compound. So what is a traditional natural product workflow? So you have your raw material, you do the extraction using liquid extraction, or you do solid liquid extractions using the Soxhlet extractors. After that, you do the purification using the glass columns. Once you have them, then you do your traditional TLC. But then things have modernized, wherein you can use automated extractors here for this particular step of extraction. For purification, you can use cartridges, or there are automated systems which will help you for the purification part. And when it comes to the TLC, you can use an HPTLC spotter, and this, if coupled to a mass spec, give you very good information. So this is how it helps. So when you have an HPTLC coupled with mass detector, that you have your natural products, you do the filtration. Okay, if after doing the extraction, you do the filtration, you load it onto the HPTLC sample loader, you develop those plates. Once you have those plates developed, you have the CAMAG TLC interface, wherein your bands, they are sucked in, and once they are aspirated into the mass detector, you get the molecular weight information also. So this is how you get your samples extracted, your analytes being separated, and after your analytes are being separated on the TLC, you get their confirmations using the mass detector. So HPTLC applications within natural products. So they help in research, they help in drug discovery, they help in regulations, because the regulatory surveillance, the standards and the guidelines, because you are more confident in saying that, okay, this is my target compound because the molecular weight matches to your target compound. It helps in manufacturing also, in better product development, and it also helps in QC testing. So these are the various customers who are using such a setup. So most of the natural product application, majority of the pharmacopoeial methods relating to natural product raw material analysis, they refer to TLC or HPTLC. But what is the challenge when it comes to such analysis is that you have overlapping band on TLC plates. You have faint bands which are not so visible onto a TLC plate. There is a lack of reference standards because you might not have reference standards available readily for all the bands or the analytes that you are separating. And accessible mass detection can help with these challenges. So if you have overlapping peaks, not an issue, because with the QDA, if they have different masses, you will see them as different mass peaks in the mass spectrum. If you have faint bands, then also they would still have the molecular weight, and hence you will still see their mass MS peak. If there is lack of reference standard, that is also not an issue because of the molecular weight information being readily available, you can say that, okay, this is your target compound of interest. So why mass detection to an HPTLC? Because it's too complex and not robust enough for HPTLC analysts. Reference standards are not often available to confirm the band identity. Confirmation of overlapping bands on plates. If there are faint bands on the plates, they can be difficult to identify, and hence that becomes too expensive. With the QDA is a true mass detector with user interactions based on PD and TUV, enabling performance across the user levels. There are minimal opportunities to go wrong because the tuning happens automatically. It's readily accessible to the user. The ESI is also pre-optimized, and the ESI probe is also a single piece. So this becomes very more robust platform to use. There is an excellent uptime of QDA is proven in this challenging environment, because even if there is some maintenance, the researchers or the scientists can do it themselves. The QDA can be coupled with HPTLC, reduces difficult sample preparation issues, thereby allowing the botanist or phytochemist or the pharmacist to try its samples for high quality HPLC or UPLC analysis. Mass selectivity can assist in identifying the unknown peaks without reference standards, confirming overlapping analytes, and identifying faint bands. This increases analytical certainty and improves lab efficiency. Because lab efficiency, my friends, today when we are working in, say, an institute or the university, we might not realize it. But then when we go into the private world or in the private industry, efficiency and productivity becomes really important. And hence, such technologies would help you to become productive, not just now, but even later on. So this is how the workflow is with all the modernization which is now available. So you can do the sample application using the sample loader. You can have your chromatographic development in the chambers. You can have derivatization chambers. Then you have these detection systems wherein you can visualize the peaks. After this, then you can have a hyphenation of the TLC. So you have your TLC-MS interface. This then get coupled to the QDA, and you can get the molecular weight information also. With this gel doc, what you can also do is you can do the documentation. So you can document your results as well. So this is how it goes. How it actually works is that you have a HPLC pump which goes at a flow rate of 0.5 ml. This is coupled to the TLC-MS interface. Because of this, your bands, they get aspirated and they are taken into the mass detector. So this is the QDA, which is from waters, and this is where the electrospray ionization happens. So the TLC-MS2 interface in flow path between isocratic pump and the QDA. User manually positions the plate using the laser cross heads, and elution head is lowered. So you are placing your plate here and you are lowering this head. Valve is switched and the solvent flows through the zone on the TLC plate. So this is where your solvent from the HPLC pump is coming in, and the sample flows directly into the QDA for analysis. So this is where, as your sample input is going in, through the sample output which is happening, the filter located inlet at the 6-port valve before the eluted zone gets into the MS. So this will prevent the silica which is in there in the HPTLC plate from coming in, and hence this allows only your analytes or your analytes to enter the mass spec. So this is how it is, that from the sample in solvent input, your solvent comes in here. This is where it gets dissolved in the TLC, in the HPTLC plate, and then you pick up this band and it goes directly into the QDA mass detector. So this is how it is. So you have your HPLC pump which delivers the solvent. Pressure control piston is lowered, so you lower down this piston, and then HPTLC plate with the substance zone. So this is your band which is there, and the mass spectrometer then analyzes the sample and you get the molecular weight information. So once this is done, then you can move it on to the next band, and then you can get the molecular weight about that band as well. So then the MS interface has round and oval elution head. So depending upon the type of bands that you can see, you can use the type of heads that you want to use, and hence you can get further informations. So here you positioning of the plate with the laser on the specific spot. So you can spot, okay, this is the position that you want to aspirate. The piston lowers with 4 bars and the elution starts. Compatible software for TLC MS interface 2 with QDA. This configuration is compatible alongside QDA supported versions of MassLinks and Empower. The KEMAC is manually controlled and requires no software interface. So the KEMAC system, which is the MS interface, does not require a software interface. This configuration can be controlled by MassLinks as well as the Empower software. So what are the type of solvents? This is important because if you're working for positive ionization, then these are the eluents or the solvents that you can use. When it comes to negative ionization, these are the solvents again that you can use. Once you get the results, it is important to expect that there would be some sodium adduct or some potassium adducts to be formed, and hence depending on this information you can do further mass spectral interpretations as well. So these are the steps that first you do the rinsing, then you have the first blank, then after that you do the rinsing again, then you take the second plate blank, then again you do the rinsing. So this will basically help to do the background correction, and after that you can have your substance measurement which can be done. So you do the substance measurement subtracted by the plate blank, and hence you will get your final results. So you do the measurement of the system, you do the second plate blank, you do the substance measurement. So then after that you have to subtract the blank from the substance, and hence this is your final data. Because what we see is that the blank portion or the background itself will have some spectra, and hence that spectra has to be separated from the substance, and after doing that you get the final results. Now when we talk about applications, there are many application notes focusing on reaction monitoring, dietary supplements, cannabinoids, adulteration of natural products, and cosmetics. So we'll just go through some of them. This is about adulteration in natural products. So this particular plant part, which is St. John's wort, is widely used or it is sold as a supplement in mass market. So there is many types of adulteration which is done. So here what we have done is that we have separated 4 synthetic dyes using the HPTLC, and using the TLC MS2 interface, we directly elute the target zone from the HPTLC into the QDA mass detector. So you can say that this is the standard and then this is the suspected. Then hence you can say with confidence that whether your sample has been adulterated or not adulterated with that particular dye. So this can be used for adulteration. This can be also be used for detection of sibutramide in slimming products. So diet products these days are again in demand, and hence in them also if you want to identify the presence of certain compounds, then that can also be done. So here again we have done the HPTLC separation. After the HPTLC separation, we have further confirmation of the standard with the slimming product, and we can say that this is the peak of interest and this is what is present in the standard as well as in the sample. So this is important because with the QDA, especially for samples with low concentration of the adulterant, even those can be detected using the QDA mass detector. Next, if you want to do some in-process quality control, because these days people are not just looking at raw material or the final product, but if they try to optimize the process itself before the final product being generated, the TLC MS can be used in process control also, so that if in the process itself, if you want to do some optimization, then you can do the optimization as well. So this will provide you reliable analytical endpoints and significantly improve the separation. There is structural confirmation that can be achieved using the TLC MS hyphenation. This method is suitable for identification of synthesis products. The ChemEC TLC-MS is used to directly elute target zones into the QDA mass detector. So you can do such in-process monitoring and optimization as well. So this is the type of analysis that we just seen in the previous slide. So with this, I thank you everyone for their patience and their participation today. Thanks a lot.
Thank you, sir.
Thank you.
For guiding us with respect to the basic instrumentation in HPLC MS system in stepwise and systematic manner.
Yeah.
Sir has also explained us various applications of QDA HPTLC in academics, industries like cosmetic industries, natural products, and food industries. Now the session is open for the discussion. There is one question in the chat box.
Yep.
From Sudipta Roy. How to measure molecular weight and lambda max for any compounds like natural products or any complex molecules?
Okay, so in this, that can be done like you have your, like we said, you can have your LC, which can be connected to the, say the UV detector, and followed by the UV, you have the QDA mass detector. So what happens is your separated compounds, which are coming out of your LC, they will get detected or analyzed in the UV. So you will get the UV spectra for those compounds. And after that, you can have your QDA. So that with that, you can get the molecular weight information also. Unfortunately, the videos didn't play. So the videos will give you very good idea about how such analysis can be done and how they would help in natural product analysis.
Thank you, sir. At the same time, there are very nice comments in the chat box. Like the session is very informative. The participants are asking for the presentations.
Sure.
And contact details.
Sure.
I'm very much sure, sir, this session will definitely help our students as well as faculty members to improve the practical skills. Thank you for your presence and expecting the same in the future events.
Sure. That will be a pleasure for us to be a part of this. Thank you a lot.
Now I request Dr. Oja to kindly accept a virtual bouquet as a token of gratitude. And also a certificate as a token of appreciation.
Thank you. Thanks a lot. Thank you.
Thank you, sir.
Thank you.
Now I request Rupali madam to kindly proceed with the valedictory function.
Thank you so much. Thank you, Dr. Ritesh Oza, sir, for enlightening us on mass spectrometry along with its basic principle of working and applications of HPTLC and MS in pharmaceutical research. Sir also explained sampling techniques like electrospray ionization, also elaborated automated startup and calibration and quadrupole analyzer theory. Sir said truly that researchers can transform their research workflow with mass detection, whether it is a synthetic work or analytical work, with more accuracy and reduced timeline. Explanation using case study was really impressive, sir. Thank you so much. It is said that the end of a story is a new beginning for many others. On this note, let's conclude a remarkable, memorable, and knowledgeable technical session. On behalf of the institution, I sincerely thank the dignitaries for being with us and enlightening us with their enthusiastic remarks. I shower my heartfelt gratitude to the professors and everyone of the institution for their wholehearted support rendered in the success of this webinar. Thank you so much. I request coordinator Dr. V.V. Chopde to give vote of thanks.
Thank you, madam. Thank you, Rupal madam.
Yes, sir.
There was excellent session hosting by you. It is a really remarkable by our honorable principal sir. Friends, now we are turning over the last or end of the session. Before that, I would like to say something to you, friends. After the vote of thanks, everyone have to switch on their cameras. We'll have the group photo. So as a memory of this webinar. Then second, one attendance link and feedback link has been posted in the chat box of the Google Meet as well as the description box of the YouTube. All participants are requested to fill that. That will help to get the participant certificate. So we'll have the auto-generated certificate systems. So within 3-4 days, definitely you will get a certificate on your email. Also, meanwhile, we'll send one certificate link to you on your personal mail ID as well as our Telegram group. Okay, that will also helpful to get a certificate to all participants. Okay, so before saying to the vote of thanks, I would like to call few participants regarding the feedback of the session. Okay, so I'm requesting to Padmanabh Mukhekar sir. So please share your views regarding the seminar and the webinar. Padmanabh sir is there?
Yeah. Thank you, sir. Good afternoon, everyone. I'm Padmanabh Mukhekar. I'm working as a product specialist in waters for emerging and growing markets. So thank you, Mr. Monty, sir, for his excellent presentation on Empower and how Empower software can be connected to the other vendor system also. And this is very user-friendly software. And I hope academic institutions and research institutions will get a definitely benefit of it. Thank you to Dr. Ritesh for his excellent presentation on HPTL CMS, where he truly explained how happened techniques can be utilized by nutraceuticals and natural product domain. So this TLCMS is a versatile technique for the separation and identification of pharmaceuticals and phytoconstituents. The benefit of HPTL CMS will be we can save the solvent where we use maximum solvent in HPLCMS, that is LCMS. And this is a versatile instrument that can be used for isolation of unknown compound like phytoconstituents and some herbal formulations, and that will help us. Thank you, sir, for the opportunity, and I am thanking from all India Water India team for providing us this online platform to conduct our presentation. And very special thanks to Dr. Ritesh for joining this session at very last moment due to some medical emergency of previous. Thank you, sir. Thanks a lot.
Yes, sir. That's very truly, sir. There is special thanks to the Ritesh sir. They have been joined a very short time. Now I'm requesting to Devgol sir is there. Uday Devgol sir.
Yes, sir. I'm there,
Hello?
sir. Yes, sir.
Yes, sir. Sir, I'm requesting to you kindly share your views, sir.
So it was very nice section. What it is, what new things are going on. We are there from very old days. So we are now knowing only the very old technologies. So we must upgrade ourselves for this new technology. So therefore I was eagerly waiting for this seminar, and we are very much thankful all the presenters that they are enlightened us with the new technologies which are there in the industry. So definitely it will help our academicians and researchers for this. And I am also thankful to you, sir, that you are arrange such type of the informative sessions for all. Thank you very much, sir.
Thank you, sir, for your nice words, which will be encouraged to us to arrange this type of webinar again and again. Thank you, sir.
Thank you.
Valmik Gite sir is there online. Valmik Gite sir from Razur. It is Arts and Commerce, Arts and Science, Canterbury College. Gite sir is there. I think he may be okay. No issue, sir. So now we are ending the last session of this webinar. That is a vote of thanks. It's a duty and moral responsibility of the coordinator to say thanks to everyone who have been participate, who have been supported, who have been done the efforts for successfully conduction of this webinar. So honorable respected Principal Sir, ex-Dean, Faculty of Pharmaceutical Sciences, Savitribai Phule Pune University, Pune, Dr. Respected P.D. Chaudhary Sir, today's chief guest of the webinar, Mr. Prakash Monty Sir, Senior Marketing Manager, Waters India Private Limited, Mumbai, Dr. Ritesh Oza Sir, Senior Product Specialist, Department of Mass Spectroscopy at Waters India Private Limited, all participants, ladies and gentlemen. Friends, it's dim and a great honor to privilege the proposed vote of thanks on the behalf of Progressive Education Society, Modern College of Pharmacy, Nigdi Pune, to a great resource person, delegates, and all organizing committee member. Okay, on the on the occasion of one-day national level webinar on Waters QDA mass detectors with CAMAG HPTLC MS interface technique for pharmaceutical analysis on 2021. A big thanks to Water India Private Limited, the entire team, for moral support for as well as the laboratory support for conduction of this webinar. I must thanks and mention our deep sense of appreciation to all resource person. Today's Mr. Prakash Bonti sir, Senior Manager, Waters India Private Limited, as well as Dr. Ritesh Oza sir, Production Specialist, Water India Private Limited, Mumbai, for delivering the very impressive, effective, informative, valuable, as well as very energetic session on this national level seminar. Sir, definitely this seminar will be beneficial for us, for our MPharm students, PhD scholars, as well as the teachers. Those are interested to work on the chromatography techniques. I'm very much thankful to Padmanabh Mukherjee sir. He's a product specialist, Water India Private Limited, Mumbai, for his great support to organize this national level webinar, because sir is in contact from last 20 days, and sir have given a lot of support. Sir have given the very good platforms, okay, to enrich and encourage the knowledge. I may like to express my sincere thanks to Dr. G.R. Ekbote sir, Chairman, Progressive Education Society, Professor Shyamkant Deshmukh sir, Secretary of PE Societies, Mr. Inanda sir, Deputy Secretary of the PE Society, all Management Council members, for given permission to arrange this type of webinar at our research institute. I wish to express my gratitude to beloved our principal, Dr. P.D. Chaudhary sir of this college, for providing the required facility, always encouraged to us, and logistic support for organizing this type of webinar to all committee members. I would like to take opportunity say heartily thanks to all delegates and their active participation in the webinar. I am thankful to all principals, HODs from all over India who have deputed their faculties as well as the research scholars to attend this webinar. I am thankful to Dr. Padmaja Khore ma'am, Professor Chitra Patil ma'am, for smoothly handle the registration committee and the receptions. For all delegates, how to join, they have provided the link and everything by mail or webinar, Telegram groups or WhatsApp groups, time to times. Thank you, ma'am. I am very thankful to Professor Vishwakar sir, Rohini Chauhan ma'am, Anjali Kumar, Shubhda Jagdale ma'am, and their entire team for giving the very excellent technical support. Friends, as we know, without technical supports, it's very difficult to organize a virtual online session. And these technical teams are working from last 3-4 days continuously. They have made the arrangement of the Google Meet as well as the creable. Okay, I'm very thankful to you for smoothly conduct the sessions. Last but not least, I'm very much thankful to all teaching faculty, office staff, non-teaching members of the Modern College of Pharmacy for their involvement and willingness for successfully completion of one-day national level webinar. So once again, I'm very much thankful to you, and I hope we look forward continuous association with you in the future. So I'm thankful to you. So I'm requesting to all of you, so kindly switch on your videos. So we'll take a screenshot as a group photo, as a token of memories. So I'm requesting to all, please switch on your videos. Anjali madam, kindly take a screenshot, which will be really remarkable for us, because from morning, Ritesh sir and Padmanabh sir, Prakash sir also join with us, because they are an industry person. They will be very busy. Still, they have accepted our invitation and they spare their valuable time with us without any expectations. I'm really thankful to him.
Yes, sir, we have taken screenshot.
Thank you, madam. Thank you very much. Now I can declare that on the behalf of all organizing committee member, I'm declare that this seminar is over, and I'm requesting to all participants kindly fill the attendance link, kindly fill the feedback also. Okay, so which will be very helpful to give the attendance participant to you. Thank you, sir. Thank you, Vijay sir. I'm very much thankful to you, sir, because without you, it was very difficult to successful of this webinar. Because as our Ketan Sawant sir was suddenly medical emergency was happened with him, and you have really done a very good job, sir, for this kind of webinar. Thank you, sir. Thank you once again.
Pleasure,
Yes,
sir. Thanks a lot, sir.
Yes,
Thank
sir.
you, sir.
And hope so, sir. This pandemic situation will be controlled very soon, and will be meet physically in our college.
Sure.
All participants and all our speakers are very most welcome in the research center of Modern College of Pharmacy, Nigdi Pune. Thank you, sir. Thank you.
Definitely.
Okay, sir.
Thank you, sir. Thank you.
Okay.
Sir.
Yes. I request to all technical team, you can end the meeting. Thank you.
Thank you so much. Meeting will be ended. Thank you so much.